All right, so to recap, well, we've gone through, remember we're talking about the cell cycle here, right? Cells need to duplicate. And so the way they duplicate is they go through the cell cycle. The cell cycle is made up of two stages. One is called interphase, which we talked about in the previous video. And now we're going to talk about mitosis, where the actual splitting occurs. Remember, mitosis, just to remind you, before mitosis, the one thing that has duplicated is the DNA, so that each cell gets a copy of the DNA. So mitosis occurs in what we call somatic or body cells only. It doesn't occur in reproductive cells like sperms or eggs. So every part of your body except your reproductive cells, like for female sperm, eggs, and then for male sperm. Every part of your body, so your skin cells, liver, heart, they all undergo mitosis. So mitosis is the division of the nuclear cell and its contents. And so here's an example, but we'll go through each part. So just, some people use this term PMAT. You might hear it. So PMAT stands for the four stages of mitosis. So you have P, which is prophase, metaphase, A is anaphase, T is telophase. So we'll start with prophase. So prophase, the DNA condenses in the chromosomes, nuclear membrane breaks down, releasing the DNA. So if we were to draw this real quick, I'll do it in yellow. Here's your animal cell, right? And it's made up of a nucleus, but the nucleus starts breaking up. So I'm going to draw it like this, and then what happens is your chromosomes, which I'll make orange, they start condensing. So you get these chromosomes. Normally they hang out, but they don't condense, like they're not stuck together. Normally in a normal nucleus, so let me just draw this is what it was before. It goes to here. It's the nucleus you had here, and then your chromosome is kind of just like everywhere. It's just like that. And then so it starts condensing. All right. The next step is called metaphase. So what happens is the chromosomes line up in the middle of the cell, and then the spindle fibers form at the poles. So what happens is they start lining up. Remember, the nuclear membrane is gone. They start lining up. But out here, I'll put them in green. There's these fibers, and they start forming at the poles. And so what happens is they start, it's almost like fishing. So imagine like the chromosomes are a fish, and so these spindle fibers are kind of like the fishing line, like that. Okay. So the chromosomes, they line up in the middle of the cell, and then the spindle fibers, they attach to the chromosomes. Next one, they're ready for anaphase, so the chromatids separate and move to opposite sides. So let me just do. So just to remind you, I'm just going to do a close-up of 2 chromosomes. Now, remember, you have 46 chromosomes, and this happens, but I'm just going to, just so it's easier for us to see. So I just made 2 chromosomes, and you have the spindle fiber. The spindle fiber attaches here. And so what happens is, I'm going to draw another one. Now let's just double back real quick. So remember, a chromosome, just to remind you, I'm just going to put it off to the side. So a chromosome is made up of 2 chromatids, right? So if you forget, watch the first video. And so here's one chromatid. So this is one copy. And then the other copy is here. So this is the second copy, right? Remember, because in the S, during interphase, during the S part, the DNA duplicates. There's now two copies. So what happens is the spindle fiber actually pulls apart. So here, it pulls apart. It separates the chromosome. So now you have one chromatid here and the other chromatid here, right? And so now it's good though, because remember these are copies. So here for example, like if we drew this is copy number 1, and then this is copy number 2. Right. Okay. And that's good. We got to separate the two so we can make two separate cells, but they're going to have the same copy of DNA. All right. And then last is telophase. So what happens is you've brought the spindle fibers have brought the chromosome. So the spindle fibers have brought the chromosomes to each side. So like if we had 4, actually let's make it, okay, so they brought them to two sides of the cell. And then what happens is, so that's what happens, the nuclear membrane starts reforming. So now you can start to see there's going to be two cells, right? Because now you have two nucleus or two nuclei, and then in cytokinesis you basically pinches it off. So in cytokinesis, let's draw it. So what happens, actually, now you have, I'm going to draw the two nuclear membranes, right? So what happens is, you now have two nuclei, right? And then so the cell starts splitting, it starts pinching off. So that eventually you'll get two cells with a nucleus, and I'm just going to color this orange because that's where the DNA is, right? So you can kind of see. So cytokinesis is the actual pinching part, is where the cell actually starts, the cytoplasm starts pinching into two, right here, it starts pinching into two different cells. Okay? So that's it. So let's do the chromosome number. So before the cell cycle starts, you have one copy of DNA, 46 chromosomes. And then during the S phase, the DNA replicates into 2, right? So, and then after cytokinesis, each daughter has one copy. So you have one cell. Let me go back. So you have one cell with one copy of DNA, and then now you have one cell with two copies, and then after you split, after mitosis, you have one copy and one copy, right? And that's what you want. You want a cell with a copy, and so that's the whole point of doing the doubling the DNA, is that each cell should have its own DNA, own copy of DNA. This is a really good video if you want to review what we just watched, okay?